Hello friends, today we are going to study about the cell cycle, which is the most important part of the cytogenetics. Myself, Dr. Asha Narhe, Assistant Professor, Department of Botany, Changu, Kana, Thakur College. Today we are going to study about the cell cycle. So the definition of the cell cycle is nothing but the sequence of event by which a cell duplicate its genome, synthesizes the other constituent of the cell and eventually divide into the two daughter cell. It is termed as a cell cycle. So when one cell decided to divide, it have to uh, goes into the n number of the sequences, n number of the events where their cell organelles, their nuclear structure, they are getting duplicated and finally they are divided into the two daughter cells. So it is the cell cycle. So discovery of the cell cycle, first this cell cycle is described by the Howard and Pillay in a 1953. And in a simpler terms, if you are define the cell cycle, it is a series of events involving the growth and division of a cell from the time of its formation to its own division into the daughter cells. It is nothing but the cell cycle. Now, the duration, or you can say generation time of this cell cycle, is the period between the two successive cell division, or from the beginning of one cell division to the beginning of the next one. Then factors affecting generation time. It is a type of a cell. So which kind of a cell it is going to divide. So whether it is a mammalian, whether it is a prokaryotic, whether it is a eukaryotic cell. Then the environmental factors such as oxygen supply, food availability and the temperature, they are also responsible for the generation time of the cell cycle. So here are some examples of generation time or the duration of a cell cycle. So a bacterial cell divide every 20 minutes because they are reproduced after 20 minutes. So their cell cycle duration or generation time is a very less. Then each cell divide approximately every 90 minutes. Mammalian cell divide roughly once in a 24 hours. Then plant cells such as onion root tip cells may take around 20 hours to complete a cell cycle. Then majorly this cell cycle involves two main phases. First one, interphase, which is a very long process or the long phase of a total cell cycle, which is around taking 70 to 90% of duration time of a whole cell cycle. And another phase where the actually cell get divided into two, which is known as M phase or the mitotic phase, which is a shorter phase of nuclear and cytoplasmic division. Then interphased, it is a preparatory phase between the two successive cell division, also known as a dividing stage. The interphase is sometimes mistakenly called as a resting phase, but it is actually not a resting phase. It is actually intense synthesis and the growing phase of the cell, where the cell actually grows, then their DNA get duplicated. So it is a synthesis and the growth phase of the cell cycle. Interphase, in the particular interphase, the processes such as a synthesis of nucleic acid, that is a DNA and RNA, with a nuclear proteins such as histone proteins, then duplication of the centriole in case of animal cells, then the energy rich compounds which are required for the mitosis and the protein which are the needed for the spindle formation at the end of the interphase, they are synthesized in the interphase. Then as I previously said, interphase takes up to the 70 to 90% of the total time of the cell cycle, lasting for the 10 to 20 hours. This interphase is divided into the three stages. First, G1 phase, second, S phase, third, G2 phase. In a G1 phase, the first growth phase, or it is also known as a post-mitotic gap phase, where the particular cell grows after division. Then the S phase, which is a synthetic phase, so where the actually nuclear, that is RNA and DNA get synthesized, and G2 phase where the particular second growth phase, pre-mitotic gap phase. So interphase divided into these three, G1, S and G2 phase. So first G1 phase, post-mitotic gap phase, so where the cell grows in a size and synthesizes the RNA and protein only. Then the nucleotides, which are the important for the DNA synthesis in a S phase, as well as the amino acid, they are produced in the G1 phase. So cell organelles increases in a number, cell organelles such as Golgi bodies, then endoplasmic reticulum, in case of plant cell, chloroplast, mitochondria, they are increases in a number. But DNA synthesis does not occur at this stage. The cell may enter, some of the cells, they may enter into the G0 phase, which is also known as a quiescent phase. So if the particular cell is not going to divide further, it goes to the G1 phase. But some of the cells are completing their resting period in a G0 phase, and after they are again start their cell cycle. Once the cell passes through the G1, it enters into the GS phase, and this G1 to S phase, there are checkpoints, which is a cyclin and a cyclin dependent kinase, where they check the particular cell growth properly, and then only they can allow to pass into the S phase. Then synthetic phase, or it is known as S phase, where the DNA replication takes place along with the synthesis of nuclear protein, histone proteins. Then chromatin fibers duplicated to form a sister chromatids connected by the centromere. Chromosomes carry a duplicate set of genes. DNA amount doubles from diploid to the tetraploid in a diploid cell. So synthesis of DNA, that means the diploid cell, diploid nuclear material getting again double. Then in case of animal cells, centrioles in the centrosomes, they get duplicated to forming a two pairs of the centriole. So this is a process takes place in the S phase, that is a synthetic phase. So DNA replication takes place. Then third phase, that is a G2 phase, or which is also known as a second growth phase or pre-mitotic gap phase, where the protein for spindle fibers and aster formations are synthesized, where the actually RNA synthesis continues in a G2 phase also, and the cell organelles are multiplying. Then the energy molecule, that is the ATP molecule, are synthesized and stored for the mitotic phase. So you can see here, so this particular interphase, from this, after completing M phase, it started the cell cycle again. So G1 phase, S phase and G2 phase. So from G1 to S phase, cell enters where the some checkpoints are there. From S to G2 phase, again cell goes into the G2 phase, but through the checkpoints. And again, after completing G2 phase, that is the third phase of the interphase, again there are the some checkpoints are also there, whether the particular cell are having a correct number of chromosome or not. If they having lack, or they having any kind of any changes, so that cells are not going to M phase. Then M phase, which is known as a mitotic phase. So the M phase begins after completing the interphase, and which include the nuclear division, which is known as a karyokinesis, and followed by the cytoplasmic division. So karyokinesis, which are ensures the orderly distribution of duplicated chromosome to the two daughter nuclei, and this karyokinesis involves the four phases. First one prophase, second metaphase, third anaphase and telophase. So for nuclear division, the cell goes through these four phases: prophase, metaphase, anaphase and telophase. And at the end of the telophase, the whatever the duplicated chromosome, they are separated into the two daughter nuclei. And after finishing karyokinesis, cell goes for the cytokinesis, which is known as a cytoplasmic division, or it is also known as a cytokinesis. And after completing a cytokinesis, this particular separation of the two daughter cells finished with. Then there are two types of cell division. So M phase can be for somatic cells, there is a mitosis. For the reproductive cells or the gamete, there is a meiosis. So mitotic or somatic cell division, which is a characteristic division of the somatic or the vegetative cells, where the one parent cell divided into the two daughter cells. And meiosis, which is a characteristic division of the reproductive cells or the germ cells, in which eventually gametes are formed. From the one parent cell, it goes to the meiotic phase, meiosis 1, meiosis 2, and finally there are 4 haploid daughter cells are produced. It is called as a meiosis, or it is also known as a reductional cell division, where the particular nuclear get half. Thank you.